All right. Hi, everybody. Today is sort of about looking at primary lit, how to cite, differences, but mainly kind of running around the interface here. So first step for anything in any sort of, as we're doing this lab, but again, this is approachable for anybody on the internet. If you found this video, hooray. It's just a quick guide. Hopefully it's translatable. But first step into getting to know anything and to create a project or to start thinking about what you would do, you got to learn about the field a little more deeply than the interest. Like I have interest in plenty of things, but if I wanted to do a real project or like an experiment, we'd have to find where people like kind of stand on the shoulders of the others. So Google Scholar, hooray. There are plenty of ways to search lit. You can do it at the repositories themselves, things like that. Google is, again, not to, not to show off all my Chrome and everything else that we got going here, but it's pretty comprehensive. It's not bad. So if you wanted to start something with dentistry, you would immediately go in and search here and use the terms that you would use in a typical Google search and say like dentistry cavities. Oh my God, I misspelled in my first video. Next, you would like run through these. You would like start clicking things. A lot of these are going to, you'd actually probably want to narrow down your search and say like, well, medication, like what medication treats cavities, things like that. You would keep going. Custom ranges for age as well. You will find that a lot of papers in Google Scholar, the first ones that come up first, they're not always technically going to be the most recent. The algorithm is going to give you what it thinks is the most relevant. Again, it does not mean that that's a bad place to start. But you're also going to see some great metrics on here. One thing to look for, any good article should have about, I don't know, given per year, hopefully like a citation or two per year. That's kind of the hope. The fields change with that. But for the most part, you can use this sort of as a filter. Like when you can see how many people are cited, like on this one, like a ton, pretty reliable then. You can sort of, and I'm not saying don't look into it for yourself, but using the wisdom of the crowd, typically a good place to start, especially when you're at the undergraduate level, like we're doing this tutorial at. So let's get into something I like more. DLBCL. Let's go there. Diffuse large B-cell lymphoma. That's what I do. So if I just Google that immediately, even me, I mean, I've been in the field for closing in on like 8, 9 years. I mean, some of these seem fine. Like I would need keywords that would narrow things down quite a bit. But if I wanted to go through here, I could see that even just by the language on some of these, first definition that I kind of want to show you is there's primary research and there's secondary research. Primary means data, collection, figures, stats, the whole thing. Secondary is going to be like a review article. So primary is going to be like this example right here. Let's say best DLBCL article ever. Okay, I'm going to click that and look, this is great. Look at that guy. He's right there. Anyway, now in this case, this will take you to articles. And what I want to show here is this a good example of primary articles. But in this case, my own article on here is actually under lock and key, sadly. So I wonder if I just do R2 chop harder. Yeah, there we go. I mean, I obviously have it. But primary article that you can find on Google is going to have several features that distinguish it from a review article, which is more like, here's how to get to know the field in a very detailed way. In this case, most of these are going to have a massive author list. An abstract will be a little more technical. The introduction is going to be pretty like segmented. The key things, though, that a primary article is going to have, you're going to have materials methods. Review articles don't actually do anything, right? They're not actually like functionally doing anything. These will. Look for stats, look for numbers, things like that. So looking good here. We're obviously going to do some more. We'll probably use this as an example later, but like how to make a table. You can make something like this in Adobe Illustrator or in PowerPoint really easily with some clear lines, some thickening, some numbers, and that's about it. This will be we'll come back to how to do tables later, but you can consider this like a decent start right there. Obviously kind of shooting from the bias hip there, but hey. Speaking of primary article features, results, data. Review articles technically don't generate data. They're just kind of summarizing what we know. Figures should have numbers. We should be trying to find something with this. That's what this article's doing right here. You got listing names, you got numbers, you got all kinds of stuff running around here. And equally, you're eventually going to get to a very specific discussion, and the typical intro methods, results discussion format will typically be in those primary articles, where a review may only have like intro and then kind of keep going on those lines. So that's a good example of what to look for. Also, you'll get your references right here. It can depend, but typically a review article will have like almost twice as many references, because they're citing all the old stuff to show you like a story of whatever they're talking about. A primary one's going to have less, because they're only going to cite the things that they're using and talking about, especially in the intro and discussion, potentially in the methods, especially if you want to cite the protocols and stuff. But it will be a shorter list, essentially. Get out of here. So let's say you find a primary article using your Google Scholar right here. Perfect. When you get to the sites, there's a lot of tools that you can use to go a little deeper. For example, if you want to learn more about the field and you find one really good paper that really fits like what you're looking for, right? Do you really want to go back into this hornet's nest right here and keep searching and slogging when it's like, this is actually a pretty good fit, this one right here, let's say? Here's the perfect thing to do. Number one, go back to that citation list of theirs. So these are the citations that this paper is citing. That's how to look back in the past on this stuff, right? I can look back here. Here's all the stuff that we cited, for example. Plenty of stuff from the old days, plenty of legendary stuff, all the good stuff. Side note for when we get to it, this is where you can start building a library of what you will cite as well. You'll find that for all the power that the internet has, the best source of a lot of science is simply reading and building on articles specifically. So I can go in the past here. I can see everything that was cited. Awesome. But let's say I want to go the other direction. If you found your article and you're like, I love this one, you go here. You find who has cited it in the future. I'm going to go to metrics right now. Right now, I can see, all right, here's all these people that have cited me. You can go to Crossref, Web of Science. Here we go. There's all these people that have cited this paper. Talking about SPEN, one of the genes mentioned in the paper, talking about subtype guided stuff. That's kind of the idea with the personalized med. Pathogenesis, some general DLBCL, diffuse large B-cell lymphoma papers. You can kind of take whatever you need from here. That's how to go into the future. So going back in here, one other key, and this is going to be key for my little bioinformatics class that we're doing these videos for, but again, these videos are going to be made for that general YouTube everybody audience, or whoever else takes the class in the future potentially. Here's another tool. There's no other way to get publicly available data than by going and finding it. So taking a look right here, let's say I want to begin a project. And I know I searched dentistry and cavities to start. You could also do those with those articles, but I wanted to be sure I could do it right here, hence the control. So leukemia is the journal right here. It has a pretty standard little approach. I believe it's a Springer journal. So there's Wiley, Springer, some of the big guys. Most of them will have sections like you see right here. There's one in very particular that I would like everybody to pay attention to, supplementary information. This is where the authors will decide, in this case, to provide the data that built the study, and obviously do supplemental figures that didn't have room in the original study, things like that. Look at all these tables right here. Basically what the authors are doing here, what we did, we're showing our cards. We have nothing to hide. And here's the interesting thing about data and science and everything. There's plenty of authors that don't have this section and they don't share anything. And that's that. And they just say, email me. Not to get outside the technical part of this video, but like, yeah, that's not great. And I get being competitive in your field. It's not ideal though. And in a perfect world, you got a bunch of stuff right here that not only me, you, everybody can build on. And there's stuff in these data that like we can analyze right now that, you know, the paper didn't do. There's so much data out there that we don't have enough people or time to analyze it. And that's what this class and that's what you listening right now can do. So if I go to supplemental table 1, let's download it. Oh no, my little thing is there. Let's open it up. Let's see what we got. Now, again, it's a bit of a gamble. It's a crapshoot. It may not always come as some gold mine. Come on, little guy. There you go. Oh yeah, there's my lab plans, dang it. So got this table set up. This looks like, in my case, I know this. These are the clinical statistics of each patient in each row, patient ID, what their treatment was, how long they lasted before an event, if there was an event, what subtype they were, their age, gender, the IPI, everything. So table one, pretty clear what it provides. We've got some good sheets down here. It's the R2 CHOP versus the CHOP, R-CHOP. Those are two lingos from the DLBCL cancer world, but I mean, for the most part, still very accessible. So perfect. Came in an Excel universal format. I love coding as much as the next person or like heavy data, but if you can get it in Excel, everybody can click it. Everybody can see your cards. It is a lot easier to say, here's how the magic trick worked. Here's where our evidence came from, than having to pull everything out after somebody has said, you know what? There were one or two things in this paper I didn't find very believable, and I looked into it and I can't find much more. When you provide as an author or as just any project manager type person, and when you provide the data, you answer those questions for yourself. You got to be truthful, obviously. And this eliminates the issue of show me what you got. We are. But the key need here is that you could take that data that we just downloaded. You could begin combining that with other analyses and build your own data and your own graphs. Let's say I only wanted to check, for example, how people over the age of 65 did on this therapy. I could take that data right there, filter it, do my own analysis, boom. It's a very simplified way to do it, but it's perfect. It's exactly what right here is what we want. So whenever you are searching through your articles, things like this, running around, let's see. Oh, this is a legend. I've clicked on a legend paper right here. Sorry, I can't talk about the video. It's too good. This is basically the one that separated, screw it. This is basically one that separated DLBCL into two distinct RNA subtypes. GCB-like and ABC-like, the aggressive ABC versus the like sort of recycling GCB. Anyway, sorry. Let's go down. It's from 2000. We've still got stuff down here to show here's the data that we have. Sometimes it's going to load in a PDF instead. But this is still stuff that you can begin to sort of use and talk about and stuff. Pretty neat. Sorry, I got a little nostalgic there saying that. So, cool stuff. That is how you can sort of run around and use Google Scholar. I want to use it one more time to just do this. If you just type the word review in, we'll most likely get a bunch of reviews. So I know this paper right here, 2019. Let's take a look. Okay, I keep getting ones that I can't access. Take a look. Let's try one more time. Again, one thing that I should have noted to begin is that most of you watching will, well, hopefully, well, a few of you will have access to a library with funds and with abilities to access unlockable PDFs. So follow through on those resources if you need. Now, we got one that has a PDF right here. Diffuse large B-cell lymphoma. This is a pretty nice little review. Oh no, is this somebody's? As you can tell, notice from the time that we had the data, the primary article, this one is full of text. There's a lot of stuff going on. There is going to be some data. And now that I'm looking through this, I'm getting a little worried that this is somebody's thesis, actually. Let me take a look. I'm going to make sure it's cited by somebody. If it's not cited, what did I just click? No, that's cited by 521. That was definitely a review article. I want to see, yeah, I want to see treatment approaches. Yeah, this is Wyndham Wilson and Stout. They are both big guys in the field, and Nurkowski. I know this one. Yeah, we're running into the main problem of the fact that a lot of these are not just accessible out of the gate. Management. Oh, wait, there we go. One should be good. PDF EPUB, current strategies, future directions. Perfect. All right, this one's a little more meta, like clinical, but perfect. So running around this review article, you can kind of tell there's a lot more overview and a lot more like sort of historical looking figures. There's not many stats, right? Not many things running around in that case. Dang, I am striking out. I should have stayed in dentistry and freaking cavities and stuff. I would have gotten a better review. Here, let's go for. Oh, okay. typed in Dr. Morin's name. He's a guy that does bioinformatics, and I rely on for a lot of good stuff. Hudson, Arthur, other great authors too. So, of course, there are articles, free access. Let's take a look. Sorry for hanging on there. Here's what a secondary article looks like, where we are talking about the field in general and review. Another key thing, there's fewer authors on a review typically because it takes less to sort of write, but see how there are good overview photo, like pictures and cartoons and stuff on here. Not necessarily data, but also enough to show, like, sometimes there might be like overview data like this. This is actually pretty data-y. Yeah, wait a sec. No, it literally says review. I can't be wrong. Good. Their reviews are just pretty good. They're kind of reviewing each of these other major articles that talked about how you use genes to classify these cancers. But again, see the amount of color overview. There's less numbers in this case. This happens to be a pretty big numbers one, which again makes it a very powerful review article. But you want to be looking for stuff like this. Really nice figures that you can translate and see exactly where each of these subtypes, for example, is coming from. So, perfect. I'm actually going to cash out of that. So I wanted to next kind of take a look at, and you can probably see it in my tabs, I want to take a look into a very easy and free tool that everybody can access. So this is Mendeley. This is an application that will allow you to make citations. A lot of you know, like, I think, what is it called? Like EasyBib and some of those other tools that like take some stuff and make a bibliography. This requires an account, it requires a download, and it requires, some time to load up some articles. But I promise it makes your life so much easier. It will do automatic bibliographies. It will do in-text citations, however you want, numbered. It'll do the whole thing. It's a monster. It's great. So what you would do is go to Mendeley. You would actually go here and find out how to download Mendeley. You would get that done, downloaded, hooray. You will end up with a profile like this and your online one. The key weakness of Mendeley, and didn't always used to be this way, but I'm kind of stuck in my ways now. You search for articles on Mendeley. So if I wanted to say, I want to know about the gene MyD88 DLBCL, I'm going to search right there. So I'm going to take a look. I've got some new stuff. I want some new stuff right here. I want this year's latest batch of articles right here. And I just did this search. This search in Mendeley is not as good or as powerful as the Google Scholar ones, who do not expect perfect results, do not misspell anything. But cool thing with Mendeley, you can get most recent, you can get the most cited ones. That's a good one because you do want to follow, like I said, wisdom of the crowd. A lot of the times it helps. So treatments, IRAK inhibitors, things like that. I actually want to go to most relevant next. TNFAP. I like this one. It's cool. So prognostic impact, MYD88 P53 mutations. Perfect. If I wanted to cite that, let's take a look. So I have my online profile here going and searching. I have my desktop application that I just downloaded, all up and rolling. Here's my giant library. How many I'm up to right now? 605. It's awesome. So what I'm going to do, I'm going to add this to the library. I like this article. I'm going to read it too. I'm not going to read it in front of you guys. I read too slow, but I would read this. Get out of there. I can read this abstract right here. I can view the PDF. It happens to be a free access article. Perfect. I can look at the PDF right now. Is this what I need? Oh no, I do not want cookies. Get out of my way. Okay. I can read through this article. And I may have already known to search for this article, by the way. I may actually have been searching and been like, I actually need this article right here. You would just copy paste the title in, find it. But let's say like, yeah, I need to cite this right now. So I just added this to my library. I'm going to do it again. I'm going to go to my library. I'm going to sync, make sure everything's there. Let's take a look what comes back up. Added, and filter that to right now. Oops, I added it twice. Well, I'm going to delete that one. Okay. Now, the next fun thing that you'll be able to add after you do the installation of Mendeley, you can actually go into Word. So, yeah, I actually think I have one of my bio ed papers, not one of my cancer papers up, but whatever. Cool thing up in here is that in this manuscript, the science manuscript right here, I'm like, all right, I need to cite something in the intro. I need to say like, all right, Mid 88 gene is bad, right? Ooh, with TP53 mutations, right? That's the article we just saved in Mendeley. What I'm going to do, once you have this little add-on in Word downloaded, this whole little cytomatic right here is going to show up. I'm going to insert citation right here. I have a massive Mendeley library, so this might take a sec to find our thing. But here's the beauty of this. You just search for it. I'm going to say 2023. There it is, right there. Isn't that nuts? Click, watch what happens. I'm going to add that right there. You could add multiple if you had multiple things to say. In fact, I totally do. I know I do. Yeah, mid 88, another P53. Yep, there's another one that kind of is older about it. I'm going to add both these right here. Watch it. Wait for it. Oh, don't wait too long. There it is. Awesome. Look at them both. Cited, set up. They're in a different format. Here's the cool thing. All the way down here, they're being built into a auto-generating bibliography for you. Everything that you need. For example, if I wanted to, oh no, where have I've lost it. There it is. There we go. So I just did Control Z to undo, Control Y to redo to find my place back. For those of you looking for fun shortcuts. Okay. Now, right now, given that I'm in one of my education articles instead, I'm actually in a like long form citation. See how it's like the whole author and stuff? Let's say that I want to go to cancer genetics. Cancer genetics is gonna, oh, I lost it because what cancer genetics does drives this. Let me see, where's my mid 88? Well, now I gotta find Control F, mid 88. It ain't gonna be anywhere else except where I just plopped it. Okay, so by going up to here to reference style, and I can download pretty much any reference style of any journal that I want. Now I just went to the little numbers right here. A lot cleaner, right? Perfect. And that's also going to change the bibliography down here, way down here. Oops, come on. There they are. Going to go down here and do the numbers. So Mendeley takes a bit of investment. I remember a good story about this was basically like I was in my PhD and writing my thing, my thesis, the whole big thing. And we were in a sort of a thesis boot camp, and our coordinator, Dr. Carmines, was like, hey, it's tough to put the citation software in there. It's annoying to get to use, but don't do it by hand. Do not try and cite by hand, okay? Don't do it. It might be okay as you're writing the first time to be like, all right, that's reference 4. That's reference 3. That's reference 7. But the minute you have to start editing and moving stuff, Mendeley keeps track of that for you. This, if you did it manually, got to be up here in some Excel sheet, and you don't got time for that, okay? And there was a person in our group that said, nope, I'm going to do it by hand. And Dr. Carmines was like, all right. And I don't think that person had a good time writing things. So keep it, use the software. That's what it's built for. It makes it easier on us. It's not cheating. That's what I would kind of say on that. So one last thing. Oh my God, do not save that because that was just an example. One last thing that I would say is, again, the key with using this is you're going to want to build a big, you're kind of going to want to build a massive like article set. So for example, I wanted this one right here. So I just downloaded him, or I'm sorry, I downloaded them. Let's see what I've got here. Sorry, my camera's in the way of myself, of my download. Okay, so I got a download right there. So what I'm going to do is actually go all the way over here to my folder and say, all right, I just downloaded this. Sorry, I'm making myself look pretty old right now by like shoveling stuff, but it takes an extra step, but downloading this and naming it. Again, sorry. Downloading it and naming it in a folder called like research articles or library, whatever you want. Doesn't take anything to download the article. Hooray. I'm sorry, schedule form. Hooray. Yeah, I'm sorry. You see my whole downloads now. So I'm going to name it usually the last name of the first author. Then I'm going to say the year, 2023. I'm going to say parentheses. I usually say what it's about. So I'm going to say mid 88, P53 in the LBCL. Okay, done. Now, I can add these as a big bulk to Mendeley, or like when the time comes, you can just copy paste the title for each into the Mendeley search. It'll come right up. You add it to the library, go to the library. The sync is actually pretty automatic. And oops, sorry. And then you go on Word and cite it. So that was the tour for today. The main stuff. Oh my God, I totally lied. One last thing. All right. When you go into these supplementary files that you're downloading, do keep track of them too. As you can notice here, I've got 22 right here. Let's take a look. So this is from data that I downloaded from a supplementary information table that I was like, hey, that looks pretty good. Take a look. I got patient stats, cells, all kinds of stuff. I got genes up in here. This is good. So when you are downloading and looking to build your project, that supplementary information, these subtables that are publicly available from the data themselves, that is your easiest access and jumping off point for real data. So that's what I would suggest, like starting with. In your first jump, in our first jump, if you're literally in the class with me, I think our goal is somewhere around like 10 to 15 papers, maybe 1 to 3 data sets that you have like found and downloaded. And we're going to start from there. Because if you're watching, you may be interested in like creating your own project idea. One of the best ways that I always remind people, and this will be my closing thought, I promise, is that if you're interested in something, think of a conflict in that field. Let's say you love animals. Let's say that you love dogs, right? What is a problem with dogs? Homeless dogs. I'm going to start looking for what is the data on homeless dogs. From that jumping point and from those articles that you will find, you will have inspiration and read and find ideas of where will I build on this? What have people said in the discussion that says, we can't do that yet, or we didn't have time? And you go find their data or use their data or integrate their data with other sources, and you begin your idea. And you say, dogs in this region or dogs of this breed do better or worse under these circumstances, for example. And voila, you have a unique integrative analysis. This is the beginning of that. So hope you enjoyed. Thanks for dealing with me running around so much like this, but I think we should be all good. You and good.